Hello and welcome to this webinar titled Subvisible Particle Identification and Characterization by Multi-Technique Methods, hosted by Biopharma Asia magazine and presented by Dr. Jonas Hoog-Thygesen and Colin Merrifield. My name is Sri Patel and I will be the moderator. Before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our presenters. Dr. Jonas Hoog-Thygesen, Area Specialist at Novo Nordisk Pharmatech, performs research within microscopy, microanalyses, and new analytical methods for particle identification. His main areas of expertise are spectroscopy, microanalysis, and advanced data analysis, chemometrics. He holds a PhD from the University of Copenhagen with process analytical technology. Dr. Thygesen is a Danish national born in June 1983. Our second presenter, Colin Merrifield, is a product manager for microflow imaging at Protein Simple. He obtained his degree in molecular and cell biology from the University of Connecticut. Before he began his obsession with aggregated proteins, he worked with solubilized ones as an assay developer for a startup in the greater Boston area. From immunoassay to particle analysis platforms, he has spent a decade helping to bring novel tools and solutions to the life science industry. I would now like to invite our presenter for the first half of this webinar, Dr. Jonas Hoog-Thygesen, to join our webinar. Welcome, Dr. Thygesen.
Thank you, Sri. And thank you for the nice introduction. So yes, I would also like to thank you, Biopharma Asia, for allowing me to speak here today. But anyhow, I am going to discuss with you some different methods for particle identification and characterization. I'm going to speak about the subvisible range of particles, but many of these techniques, as you will see, will also be usable if you have issues with visible particles. So, overall, I have three take-home messages for you today. First of all, I'm going to discuss the regulatory expectations of identification of particles with you. Then I'm going to outline how we can use several analytical techniques to obtain a synergistic effect when we do this identification. And finally, I'm also going to show you that when we use these several techniques, we do get this synergistic effect, and it will not be enough just to use one single method. So, why do we even bother with particle analysis, and why is this interesting? Well, within recent years, we've seen FDA guidance being issued where they state that we need to assess the range and levels of these particles, even in the subvisible range, from 2 to 10 microns. We need to do this both in when the product is shipped from the producer, but also over the course of the shelf life of this product. Furthermore, we've also seen a new USP chapter being issued recently on how to analyze these particles. And I'm going to discuss with you more in detail later on the content of this USP chapter. And finally, we've also seen warning letters being issued over visible particles found in production batches. And this is most certainly something that you don't want as a pharmaceutical producer. So this is a strong indicator that we need to look into particle analysis. So we've seen this recent regulatory shift towards particles and particulate matter, and this makes identification and characterization of any particles that we may see inevitable. So I'm going to discuss with you here some of the different methods that we have available. What are the strengths? What are the weaknesses of these methods? And as I've already mentioned, I'm going to show you how we can combine the methods. However, the combination of methods shouldn't be a new idea when you talk about identification of materials. If you look into the ICH guideline for validation of analytical procedures, you will see that if we have lack of specificity of one analytical procedure, we can compensate by using another supporting analytical procedure. Meaning that if you have unknown materials that you are trying to identify, then it will be very hard for you to state that this is a specific method. But if you use a supporting method, then you are still on the safe side with regards to the regulatory expectations. And these expectations are also carried on to the FDA guideline that we've seen. Here we can see that the FDA, they do expect us to use more than just one single method for analyzing these protein aggregates that may be present in the product. I already mentioned the USP chapter 1787. Now, this is a general information chapter that I find very interesting to read. First of all, it outlines several different particle types. There are the extrinsic, those are the really bad guys that you don't want in the product. There are foreign and unexpected, that could be hairs or fibers, for instance. Then you have the intrinsic, silicone, rubber, glass. These arises from sources within the formulation ingredients or assembly process or the packing material. And finally, we have the inherent particles. These are protein aggregates. So these are materials that we would expect to find either from the drug substance or from other formulation components. And the USP 1787 then outlines different strategies for identifying and characterizing these different classes of particles. As it stated, the chapter has focused on enumeration, characterization, and when possible, identification of particles. So we should, if possible, ID these particles. I've here outlined the different analytical methods that are discussed in USP 1787. Here we see the different methods and their analytical range. We see that some are within the visual range, but quite many of them are obviously in the subvisual range. The guideline furthermore divides them into different classes. The first class is related to size and distribution, determination of size and distribution of the particles. The second class deals with size and morphology. Here we will go one step further in not only sizing the particle, but actually getting a picture of what it looks like. And finally, we can go to the even more advanced characterization methods where we will get, for instance, a spectral fingerprint of the particle, enabling us directly to identify the material that the particle is made out of. Quite often when you discuss subvisible particles and analysis of these, you will hear people discussing MFI or microflow imaging or flow imaging analysis. This has developed into the industry de facto standard for subvisible characterization of particles. And as we will also see later, you can do sizing and morphology as already stated, but you can also do particle counting. The method is based on having a flow cell where you pump your liquid through. You then have optics and cameras, a light source that enables you to image what passes through your flow cell. Using these images, you could then classify them into either silicon oil, glass, air bubbles, small pieces of rubber, or protein aggregates. And this is really a powerful tool when you compare it to, for instance, the well-known light obscuration, because here you can actually visually see the particles that you have in your vial. One thing that you don't get, though, is a spectral fingerprint of the individual particle. And in order to get such information, you need to look at other types of analysis. I would therefore like to discuss some of the other methods that I've highlighted here. And I want to do this by taking a starting point in the Microanalysis Center where I work at Novo Nordisk Pharmatech. We are an ISO 9001 certified laboratory with more than 10 years of experience within the microanalytical service branch for the pharmaceutical industry. We have specialized within particle identification and characterization, and the main part of our tasks are within the visible range, but we do see an increase in demand for the subvisible range of particles. Typically, when you want to analyze such a unknown particle, you need to go through several steps to go from the unknown sample with unknown particles to a lab report that states what the particles are made out of. First of all, you need to do sample preparation. The methods that I'm discussing here requires us to have immobilized the particles. As such, we would often use light microscopy to do a first imaging of the particles, or we might actually use the light microscopy during the sample preparation. The next logical step for us would be to do either FTIR or Raman spectroscopy on the particles. Here we will get a first fingerprint of the material that the particle is made out of. Moving on from there, electron microscopy, often coupled with EDS analysis, that will allow us to get a fingerprint of the elements present in the particle, will then be the next step. All of this is then combined into the lab report that hopefully will give you a good indication of what your particles are made out of and where they originate from. I will now go on and discuss in more detail these individual steps. So the first step would, as I mentioned, be sample preparation and stereomicroscopy. We could, for instance, use a filter-aided particle isolation, so simply filtering the sample onto a filter to isolate and immobilize the particles. We could then use our optical microscope. This could, for instance, be a microscope with a double optical path, a so-called stereomicroscope. This will give us an enhanced depth sharpness, and we can then do color imaging up to 225 times, enabling us to have micron resolution. I'm going to take a starting point in a case study where we analyzed 3 samples. This sample we found 2 types of particles. The first one being long and fiber-like, and the second one being more soft and fragile. Both of them were white and had no color. From here, we would move on to FTIR analysis. This is infrared spectroscopy, as some of you might be familiar with. Here we're probing the chemical bonds present in the sample, and when we have a spectrum of the sample, we can then do library matching by reference spectra, enabling us very fast to do an identification of the particle. In this case, when we measured the first particle, we saw that this had a spectrum that were well correlated to the reference spectrum of cellulose. While when we had a look at the second spectrum, we could see a more mixed picture. We saw some peaks that originated from protein, while other peaks in the spectrum could be assigned to silicone oil left in the particle. Another possibility would be to do Raman spectroscopy on a particle. This is also based on molecular vibrations, but in contrast to FTIR, we're not using an infrared light source, but instead we're using a laser to probe the molecular vibrations. This has the advantage that we are also getting fingerprint information on the sample as with the FTIR, but in this case we don't get any signal from water, what might be a big advantage. And quite often we can go to smaller particle size when we compare to the FTIR spectra. The assessment of protein structure may be done by looking at the amide bands. Some of the challenges here is that when we use the laser, we may actually burn the sample. We may actually burn off the particle that we've spent time isolating. So this is a challenge that we need to be aware of. And likewise, fluorescence is a competing phenomena when we talk Raman spectroscopy. And this may actually drown out the Raman signal if you have issues with fluorescence in your particle. An example of a Raman spectrum would be this. We have the top spectrum here, that's an unknown particle. And then we have two reference spectra below. We have a native protein reference and a fibrillated protein reference. We see a good correspondence between the sample and the native protein reference. Furthermore, we also see that the two amide bands that I've marked here in the spectra, the amide bands of the unknown sample is located closely to the native protein bands that we have in the middle spectra. This means that we in this case would identify this particle as a native protein particle. Scanning electron microscopy would then be the next step in our chain of analysis. Here we're using electrons to image the particle. And as such, we can do very high resolution black and white imaging of the particle. Under optimal conditions, you can go up to 1 million times magnification, corresponding to almost nanometer resolution. However, when we do this, we may need to coat our sample with a thin metal layer, and this is then destructive. In the case that I showed you earlier with the fiber, we could do imaging looking like this, and we can get fairly close to the actual fiber to get a good impression of what the fiber structure is. Likewise, we could also have a look at the other type of particle that we isolated. Here, moving to very high magnification, we would easily be able to see if the protein was fibrillated, as this would be evident from the electron microscopy images. Imaging dispersive X-ray spectroscopy is then a method that we can use, since the electron interaction between the sample and the electron beam from the microscope yields characteristic X-ray photons. The energy of these X-ray photons depends on the elemental composition of the particle. Using this method, we can screen from atomic number 5, meaning boron, and upwards in the periodic table. And we can achieve micron resolution using this method. In the case study that we have at hand, we saw that for the fiber-like particle, we had carbon and oxygen, supporting very well that this is indeed a cellulose fiber. And in the case of the protein agglomerates, we could see signals from carbon, nitrogen, oxygen, and sulfur, supporting that this is indeed a protein. But we do also see a silicon peak in this spectrum, supporting that silicon is indeed present in the particle. So a short summary of the analytical results that we saw in this case study was that we had two types of particles in the sample. The first type was long and fiber-like. It had a content of carbon and oxygen, and we had a spectrum using FTIR that we could match to cellulose. And so in this case, we would conclude that this is most likely a cellulose fiber. The second type was soft and fragile. It had a content of both carbon, oxygen, nitrogen, sulfur, and silicon, and we saw spectral traits from both the protein and silicone in the FTIR analysis. So we would, in this case, conclude that this is most likely a silicone protein agglomerate. So, my take-home messages for particle analysis would be that, first of all, there are some regulatory expectations of identification of particles. Second of all, the use of several analytical techniques will give you synergistic effects, and we cannot use just one single method. We need to use several in order to get these synergistic effects. And that concludes my presentation, so thank you very much, and back to you, Sri.
Thank you, Dr. Tigerson. So just as a reminder to our listeners, if you have any questions for Dr. Tigerson or our next speaker, Colin Merrifield, then please click on the questions tab and send them through to us. And we'll be asking these at the end of the presentation. So I'd now like to invite the speaker for the second half of this presentation, Colin Merrifield, to present an industry perspective on microflow imaging as a tool for development and risk mitigation in the pharmaceutical industry. Welcome, Colin.
Thank you very much, Sri, and thank you very much, Dr. Tigerson, for an excellent perspective on particle analysis in the pharma industry. So as Dr. Tigerson presented a very broad perspective on particle analysis, my talk will be much more focused, and I'll be talking about microflow imaging. First, I'd like to present our company, which is Protein Simple, and talk about some of our philosophy in terms of bringing solutions to the life sciences industry. What we do at Protein Simple is we try to bring new approaches to the underpinning research techniques that people use in life sciences and pharmaceuticals. We've applied this philosophy to particle analysis, Western blot, ELISA, and capillary electrophoresis. And in addition to that, as you'll see on this slide, we're part of a much larger family. We're part of the Biotechnic Corporation, and that brings us in close synergy with other brand names such as R&D Systems, Novus, Clinica, Tokris. So it's really been a pleasure sharing a corporation with these other great companies. So to talk a little bit about why we're here and why we're interested in particles. This is data that I culled from the FDA's website from about, from mid-2015 to mid-2016. And if you look at the recall motives during that time frame, you'll actually see that the majority, I'm sorry, I don't know why my slides keep skipping. You'll actually see that the majority of the recall motives during this time cited particulate matter as the motive. I'm so sorry, my slides apparently are skipping. So when we talk about microflow imaging, the size range that we're talking about and that we're analyzing encompasses the 1 to 3 micron range. So this is part of the regulated particles that are included in the guidance set forth by USP, and also in the subvisible particle in the 2 to 10 micron range. That has been, that the guidance of the pharmacopoeial industry and the regulatory agency has been expanded to cover. And so a little bit about how MFI works. Dr. Tigerson mentioned this during his talk. So in the technology, we have a flow cell, and this flow cell creates a fluid path through which the sample is pulled. And then on one side of that fluid path, we have a light source, which backlights the sample. And then on the other side, we have a camera and an optical system, which is then focused on that flow path. And that provides the analysis. So that's a after that introduction to how the technology works, I'd like to talk about some of the characteristics of the system and what makes it such a valuable tool to people interested in particle analysis within the pharma industry. So the first characteristic of the system, which was touched on earlier in the talk, is its ability to produce morphology for the particles. And the reason that this is important is that new drugs have brought a new level of complexity to both the nature of pharmaceuticals and to particle analysis within the industry. I think that this concept is very well illustrated by this graphic right here. You can see the sort of simplistic aspirin molecule compared to the large and vastly complex monoclonal antibody. And with a molecule like aspirin, where the entire structure is determined by covalent bonds, you have a much more stable molecule, where the monoclonal antibody, the structure is a combination of covalent bonds, but also weaker, more intermolecular interactions, which are more vulnerable to disruption. And so previously, in a more historical sense, particle analysis had largely focused on external contamination or physical contaminants from the manufacturing line, which could contaminate the drug products. But now we have to deal with particles that are potentially a product of the drug formulation or product itself. And the morphological parameters that MFI provides can offer some good insight into what types of particles are inside of your drug product. So this is a very simplistic view of how we use morphological parameters to differentiate different particle species. So in this chart on the x-axis, you have intensity minimum, which is a measurement of how dark or light a particle is, or how translucent or opaque it is. And then on the y-axis, you have the concentration of those particles. And you can see for the protein aggregate on the right, this is a highly transparent or translucent particle. It scores very highly for the intensity minimum score. For the silicon oil drop in the middle, you have a dark perimeter with a very light center, and you have sort of a middle score for our intensity minimum parameter. And then for the air bubble, which appears very dark in the system, this score is very low on the intensity minimum metric. So by using these, we can begin to see these three different particle species differentiate themselves. And again, this is a very simplistic approach to our morphological measurements. People typically use multiple parameters in conjunction in order to really differentiate and discriminate between these different particle species. And as Dr. Tiegeson also mentioned, classifying these different particles is very important because it gives you good insight into the source of the particle and then the risk that this may present to a patient or whoever's consuming the product. And there have been categories that have been defined by the USP and Pharmacopia agencies, and these include inherent particles. So these can be particles that are formed of the drug product and its formulation, intrinsic particles that could be shed from the packaging or manufacturing, or extrinsic particles that are sort of foreign contaminants that have been brought into the drug product. And so initially, by understanding which sort of category your particles fall in, this can help determine your response to that particle characterization. For instance, if they're inherent, perhaps you want to go back and reevaluate your formulation or the stability of your molecule. If they're intrinsic, this gives you some insight into whether your manufacturing process is an issue or your packaging is an issue. Or if they're extrinsic, then maybe you need to look at the cleanliness of your lab. And these particles also offer very different risks to the patient themselves. With something like an inherent molecule, protein aggregate, this could result in an immunogenic response within the patient. Something like a silicon oil might be far more benign and offer a very low risk to the patient. and then something like an extrinsic particle could offer a risk to possibly the patient's capillaries or their pulmonary system. So really understanding what types of particles you have in your product gives you insight into the risks that your product has and what the appropriate response to those risks are. So the next thing that I'd like to talk about is the sensitivity of the system. This is another very important characteristic of microflow imaging, especially as you're analyzing proteinaceous particles. So this is an excellent study that was done by Dr. Dean Ripple at the National Institute of Standard and Technology, comparing light obscuration to microflow imaging. And so in this study, they looked at silica beads and they changed the refractive index of the matrix to study how this changed the characterization of the beads by the two systems. So these beads had a nominal diameter of 20 microns in this particular experiment. And you can see at a refractive index of about 1.47, the beads are essentially invisible to both the technologies because they're perfectly matched to the refractive index of the matrix. And then as you move to the left and the refractive index begins to deviate between the beads and the matrix, now the particles become apparent to the two systems. And in the red series, you can see that immediately MFI begins to size the beads far more accurately than light obscuration does. And this has really been an important characteristic of the technique because similar to this situation of a low refractive index difference between the particle and the solution, many protein particles are highly translucent and have a low difference in refractive index between the particle and the matrix. And this is really well illustrated on this next slide, where this is another study that was done, and protein aggregates were induced by shaking, and then the samples were measured over a time course using both light obscuration and microflow imaging. And it's striking to see that MFI actually sees far more particles than light obscuration sees in these samples. Actually, light obscuration really fails to see any sort of trend over this time frame, whereas MFI sees a clear increase in the number of particles. And these are not particles that are on the edges of the limits of detection of these two systems. These are greater than 10 microns and greater than 25. So these are highly relevant to the size ranges mandated by the Pharmacopia. So in summary, as compared to light obscuration, MFI really provides two very important attributes, and that's one, the ability to detect translucent proteins, something that light obscuration really struggles to do, and the ability to provide a quantitative particle size and shape. So that morphology that really allows you to understand the type of particle that's in your solution, and begin to understand what part of your process or formulation you should begin to focus on to reduce those particles or control them. And finally, I'd really like to talk about data quality. This is another very important attribute of MFI. And so in MFI, as in any optical technology or microscopic technology, depth of field is a very important consideration when taking measurements. And I'd like to use these two pictures of flowers to illustrate this concept. So on the left, you've got a picture that has a very narrow depth of field. So the foreground is out of focus. The midground around the tree and the flowers around the tree are in very good focus, but then the background is also out of focus. Now you could contrast that with the picture on the right side of the screen. So this picture has a very broad depth of field, and you can see that all of the flowers in the picture, whether they're in the foreground, the midground, or the background, have the same amount of focus and magnification. Now, if you compare the two pictures, perhaps you might say the picture on the left is the better picture. It's a prettier picture. It's more artistic. But if your goal is to compare all of the flowers in the picture, and I would draw a parallel between that and particle analysis. If your goal is to compare all the flowers in the picture, it's far easier to compare the flowers in the right picture than it is in the left picture. It's more difficult to accurately size and count the flowers in the left picture, and the morphology of the flowers also suffers. While the flowers in the midground are very clear, the ones in the foreground and background are largely out of focus. So this is why depth of field is a very important consideration in a flow imaging technology. So MFI was designed so that in our entire field of view, so in all the data that we capture, the level of focus and magnification is equivalent. And this allows us to more equally compare and more accurately size and count all of the particles that we gather. I think one of the key attributes of microflow imaging is the broad and holistic view that it offers of your sample. Our ability to capture 85% of all of the particles that are in the sample really offers a very complete view of what's in a vial or a syringe or a pharmaceutical product. And the ability to accurately characterize and size and count all of the particles in your sample is really what leads to a higher quality data set and the ability to make better decisions about your product. And this is something that has been borne out in the literature as well. So this is a comparison of MFI versus FlowCam and the ability to measure polystyrene spheres. These are commonly used as calibrants for the system. And you'll see that for MFI in the bottom series, especially in the 5 and 10 micron beads, not only is the sizing more accurate, but the distribution of the sizing is also far tighter. So your error bars are much smaller than you see in the FlowCam system. And this is, again, relates to the depth of field of the system and how you approach magnification within the optics. This is another example, and here you can see that even though FlowCam appears to have the better images in their system, MFI actually provides a much higher quality measurement of those particles because it's comparing all of the particles within the system more fairly and under the same conditions. So magnification is really one of the enemies of microflow imaging because you risk missing the forest for the trees. When you zoom in on individual particles, you sacrifice the quality of the entire data set. And really, again, the decisions that you make about your product are not based on individual images. They tend to be based on the entire data set as a whole. And in this publication by Genentech, this actually came out late last year, they studied the ability of the two systems to differentiate silicon oil. And they actually observed that for MFI, because of this data quality difference, it offered far greater and far more robust differentiation at all the size ranges and in all of the model systems that they used for the data. You know, they were somewhat surprised about this given the difference in image quality between the systems, but this is exactly why we designed MFI and the philosophy that went into the optics of the system. And it's a higher quality observation across the entirety of the sample and across all of the particles. So in summary, MFI really gives you a robust and sensitive insight into your sample, so you get a far more sensitive observation than you get with light obscuration. You have the advantage of morphology and being able to differentiate particles based on their observed characteristics. And really, you get the highest quality data that you can get out of a flow imaging system. That combined with the automation that we offer gives you all of that in a standalone package where you can walk away and automate your particle analysis. And I'd really like to end and thank all of our great customers, Novo Nordisk, chief among them. Certainly part of the joy and pleasure of working in this industry is working with some of the really brilliant and dedicated souls who take on particle analysis. So thank you very much, and I'll pass it back to Sri for the question and answer.
Thank you, Colin. So at this point, I'd like to begin the question and answer session. So our first question, I believe, is directed to Dr. Tegerson. You've outlined some of the advantages of using the methods you discussed. Where do you see the biggest challenges?
Well, some of the challenges with these methods that I've discussed are that they're fairly low throughput. If you compare it to methods such as the MFI that we've just been discussing, using, for instance, electron microscopy, you will not be able to analyze the same number of particles. That being said, however, I know that that currently work is being performed to automate some of the methods that I discussed. For instance, the Raman system that I discussed. I know that there's work being done to automate this.
Great. Then the next question is regarding MFI. So I think it's more directed to Colin, but Dr. Tegerson, feel free to give an answer as well. It says, is there a booklet containing pictures of known particle types identified by MFI?
We do have a particle image guide that gives some guidelines for different particle types and which morphological parameters are useful in classifying them or differentiating them within the system.
Great. Another one for MFI as well. For particle characterization using MFI, are there any benefits or considerations for using bright field versus dark field setup?
I'll be honest, I'm not completely clear on what the dark field setup is. Certainly some of the benefits of bright field microscopy are the ones that I outlined in the presentation. It's a highly sensitive technique, so the ability to differentiate particles with low optical contrast as compared to the matrix is very important. And then the ability to accurately and reproducibly derive morphological information about those particles is certainly a strong benefit of the bright field microscopy approach.
Great. This next one's for Dr. Tegerson. In the case of the cellulose particles, are there any analytical techniques used that help to identify the source of the cellulose?
Yeah, well, I didn't discuss that, but actually, cellulose is cellulose. So looking at either fibers from paper or from cotton we're not able to distinguish between those two using FTIR. But if you look at them in the microscope, so the electron microscope, for instance, then you'll often see that paper fibers are flat, while cotton fibers are more round. So looking at the actual structure of the fiber would indicate the source of the fiber. Yes.
Great. The next question, I'm not quite sure who this one's directed to specifically, but it says, can you use mass spectrometry to determine the protein ID in the protein particles? For example, is it drug substance or HCP?
Well, I can answer that one. Yes, you should be able to do so. I know that I don't work with mass spectrometry, but I know that the people who are working with mass spectrometry, they, at least some of them, do have methods for differentiating between these type of proteins. So yes would be the short answer.
Great. I think the next one, I think this is for Colin. Would it, from your analytical viewpoint, be beneficial to have a technology that gives spectroscopic fingerprints, chemical ID, together with the morphology information on a large number of subvisible particles in a protein-based formulation?
Yeah, that would be excellent. I think as Dr. Teaguson mentioned during his talk, really that would epitomize some of the synergy between the between technologies like MFI and Raman spectroscopy. I know that some of our current customers use those two techniques in close approximation to sort of gain that. They have a very broad and holistic approach and view of the sample with MFI, and then with something like Raman or FTIR, they're able to look at a smaller subset of those particles, but gain a far more in-depth view of the chemical composition, which I think is then usually extrapolated to the rest of the particles in the sample. But both being image-based technologies, there's some nice synergy between them.
Great. Not too sure who this is directed, but it says, what about glass particles? How can I set a filter method for screening glass particles?
I can take a stab at that, certainly. I think when looking at glass particles, and I assume that you're trying to differentiate them from a proteinaceous particle, looking at things like intensity standard deviation and the intensity of the particles will help to be able to differentiate those two. The only way that you'll be able to do it sort of unambiguously or in a confirmative fashion would be using one of the technologies that Dr. Teaguson mentioned, either Raman or FTIR, so that you derive some sort of chemical composition of it from them. I hope that helps.
Great, thank you. Another one for Dr. Teaguson. What method would you prefer if you were doing a HT screening of proteins?
Yeah, I'm a bit in doubt on what exactly an HT screening is, but overall, I would say that as I already mentioned once, the combination of method is really strong, and I think that also includes what Colin just said, that combining MFI with some of the different techniques that you have available, for instance, Raman spectroscopy, really enables you to get a good impression of what you're working with. So I would say go for a couple of methods and don't rely on a single one.
Great. The question, again, I'm not too sure who this is directed. It says, how does sharper images sacrifice a good data set?
So that relates to the depth of field, and what happens is that when you focus in on one set of particles or one part of your flow cell or viewing region, you lose focus in the rest of that viewing region. And so particles that aren't in that specific region that you're focused in are essentially under a different level of magnification and focus, and the image and the data quality between those two regions suffers.
Okay, great. Another one for you, Colin. How can I set a filter for screening of the glass particles by MFI? Are there available standards? Is there an available standard for glass particles?
I don't know if there's an available standard for glass particles. Sort of a common approach for differentiating two different particle types on MFI would be to create samples where you know that one sample you only have proteinaceous particles, and another sample might only have glass particles, and that gives you a really clear view of the morphological parameters that you can then use to differentiate the two. And like I said, I mean, you might look at things like the image intensity or the image standard deviation to begin to differentiate those two particle species.
Great. And another one regarding MFI. Will MFI eventually replace LO in a regulatory context?
It may in the far future. I think for now, regulatory agencies are still going to demand that the LO measurements are taken. They still want to see that the samples are within the threshold set for light obturation. I think there's more and more interest now on MFI as a supporting technology and providing insight into the morphology of the particles. Regulatory agencies want to see more complete answers of not only what particles are there, but what types of particles and where do they come from. And I know they're working on expanding the guidance for using MFI to achieve that.
Excellent. Another one for Dr. Teagerson. Of the methods you discussed here today, where do you see the biggest potential for further development? And what method would you say could make the largest impact on future characterization of drug products?
Yeah, well, I think first of all that I already mentioned automation as a really interesting field. I think that automating some of the methods that I've discussed, for instance, the Raman spectroscopy, automating that will enable it to be higher throughput, and that will, in connection with some of the flow imaging, so for instance the flow cam or the MFI, enable a much better understanding of the particles. As we already discussed a couple of times, if you have not only a number of how many particles are there, you don't only have the distribution, the size distribution, but you can actually also differentiate between the different types of chemistry in the particles. Then that's really strong. I think that's something that we're going to see more and more being looked into.
Great. This one I think is directed to Colin. It says, it's quite a large one, so bear with me. Looking at the regulations, the emphasis is on characterization, enumeration, and ID. Since protein aggregation is a persuasive problem in biopharmaceutical products, do you think the FDA and European agencies are more concerned with looking for changes in the level of aggregates in the proposed commercial product versus clinical material? In other words, correlating safety profiles. Or do they also expect to see techniques such as MFI be used at a much earlier stage, for example, in formulation selection?
I'll be honest, I don't know how much early stage development information is presented to pharmacopial and regulatory agencies. I certainly think that showing control over the level of particles, whether they be protein aggregates or from other classes, throughout the lifetime of the product is a way of proving to regulatory and pharmacopial agencies that you have a robust, well-characterized and controlled manufacturing process. And that gives them confidence that the safety profiles that you presented for your pharmaceutical are still are valid.
Right. Another one for Dr. Teagerson. Doing long-term stability studies, as we usually see, sorry, let me start one again. Doing long-term stability studies, we usually use MFI as the method for particle ID, but this method needs a lot of material. Could you recommend a different type of method in which we use less material?
Well, for instance, it sort of depends on what you want. But if you're looking for sizing of particles, I think MFI is a really good method. But if you're also looking for the identification of the particle, then, yeah, well, again, Raman is a really powerful tool. I would look into that. Perhaps filtering some of your sample onto a filter then you can use a fairly small amount and look at the material left on the filter afterwards. I've also seen people working with liquid cells in Raman systems where they actually don't filter it onto a filter, but they look in the liquid cell and look at the particles in the liquid cell. So that would perhaps be a way to go.
Great. Next one, I think we're going in 5. Is there a setting we could use? So is there a setting we should be using in MFI to differentiate protein versus non-protein as a first pass aspect ratio intensity? If so, what should it be?
That's sort of a tricky question to answer. I think that relates into what type of material you're trying to differentiate your protein from. For instance, if you're trying to differentiate a protein aggregate from something like silicon oil, aspect ratio can be a very powerful morphological parameter to use because silicon oil tends to have a very defined shape as opposed to a highly amorphous protein aggregate. But to sort of relate that back to a previous question, if you're more interested in differentiating two different particle species that have similarly amorphous shapes, like a protein aggregate or a piece of glass or a piece of rubber, then I think the intensity of the image will be a more differentiating characteristic.
Great. The next one, you mentioned burning samples with the laser in Raman. Is there a recommended wavelength and intensity laser to use for protein characterization?
Yes, I guess that one's for me. Overall, the most commonly used wavelength within pharma is the 780 nanometers laser. This will carry a risk of burning your sample, since the longer wavelength you go to, the higher the risk of burning your sample. On the other hand, if you move down in wavelength, so if you go to, say, 633 nanometers, you carry a larger risk of having fluorescence. So you have to compromise between the two. Either you run a risk of burning your sample, or otherwise you run a risk of having fluorescence. But if you don't have issues with fluorescence, I would suggest to go to a lower wavelength of whatever you have available on your system, because then you will most likely not burn your sample.
And can Raman spectroscopy differentiate the cellulose particles and the protein particles?
Yes, it can. I didn't bring the spectra in my presentation, but as we saw for the FTIR, so Raman is very similar to FTIR, as I said. We're probing the same molecular vibrations. So we will also have similar or somewhat similar spectra for the two methods. And we will be able to see the difference between cellulose and protein using this method, yes.
Great. The next one is for Colin. Is MFI fully supported in the Asia region?
Yes, it is. We have an excellent sales team in Japan, China, India, and a distribution network that supports them. So we offer all of the services that we have in North America and Europe, in the Asia Pacific region as well. Again, we have a tremendous sales function. We have field service engineers and field application scientists to help you with your work.
Excellent. And following on, what other techniques or technologies do you see as having good synergy with MFI?
I think the ones that Dr. Tigerson mentioned, Raman and FTIR, have very nice synergy, both from an image-based approach and providing the complementary information to MFI's characterization.
There's another question that's come up. What suggestions do you have to reduce the inherent variability observed in MFI across replicate injections?
That's a good question. It's sort of application sample specific. I mean, there are different things that you can do in terms of different washes between the samples, different mixing conditions, different priming conditions before the sample. So there's a number of knobs that you can turn if you're seeing variability in replicates.
Great. So, well, that looks like all the questions that we have today. So I'd like to take this opportunity to thank our presenters, Dr. Jonas Hogg-Petersen and Colin Merrifield, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com. Thank you very much, everyone.